In this chapter, we will talk about proteins, their properties, their structure, and their composition. So to start, proteins are made up of amino acids. And there are 20 common amino acids, and these we call them alpha amino acids because if you look at the structure in there, the amino group is attached to the alpha carbon. You're always going to have an alpha carbon. To the left of the alpha carbon, there is always an amino group, and to the right, there is a carboxylate group. And then on the alpha carbon, you have the R chain. It's that R chain that changes and gives the different amino acids their identity. So we call this a side chain, and this is the one that is different for each amino acid. All the amino acids, they share the alpha carbon, the amino group, and the carboxylate group. Normally, the amino group has a positive charge, the carboxylate group has a negative charge. Whenever we want to name an amino acid, every amino acid is identified by three ways. First of all, it should have a name, it should have a 3-letter abbreviation, and a 1-letter abbreviation. This is easier whenever you are writing those amino acids. So there are 4 types of amino acids. So the category of amino acids in there, they are classified into 4 groups, and those 4 groups, they are based on the R group that is connected to the alpha carbon. So the first one is the neutral or the non-polar amino acids, and those, their R chain is not polar. For the neutral and polar amino acids, this means that their R chain is polar. So as we said, it's the R chain that is different for every amino acid. The rest of the backbone of the amino acid is always the same for every single amino acid in there. So the R can be polar, the R can be non-polar in there. Now, when we say R is polar, meaning that you have an electronegative element in there, like for example, in OH. The R can be basic, and the ending of the basic is normally in the positive charge, or the R can be acidic, and the ending of the acidic group is in the negative charge. This is the table for the common amino acids of proteins. You are not responsible to memorize this table. You can have it in the exam. So as we said, the amino acids are classified into neutral, nonpolar, and for the neutral nonpolar, these are glycine, alanine, valine, leucine, and as you can see, every single amino acid has a name, has a 3-letter abbreviation. Normally, it's the 3 first letters of the name, and then a 1-letter abbreviation, and they're the isoleucine, the phenylalanine, the proline, and the methionine. These are all nonpolar because their side chain, their R chain is nonpolar. So this is the R chain over here. All of the R chains, they are made up of carbons. And this is how you know that they are nonpolar. So this is the R chain. Notice that all the amino acids, they do have the same backbone in there. This is the alpha carbon. So you have your alpha carbon in there. You do have your amino group, you do have your carboxylated group, and this is shared with every single amino acid. So over here, this amino acid is an R group. Again, whenever you see just an R group in there as a side chain, then this means that this is a non-polar side chain in there. Okay. Now, glycine is the simplest amino acid. On the alpha carbon, there is no R. Instead of the R, there is a hydrogen. This is also labeled as a neutral and a nonpolar side chain. Another classification of amino acids, they can be neutral with polar side chains. There's a big difference between nonpolar side chain and a polar side chain. For the nonpolar side chain, as we said, we have an R group connected in there. This is a disulfide. It's an S with 2 R's in there. This is nonpolar. Okay, coming over here, all polar side chains, they should end up with an electronegative element connected to a hydrogen, like an OH. Let's highlight that, like an OH in there, an NH, as you can see, an SH. This is how you know that you have a polar side chain. So that is your side chain. This is what is connected to the alpha carbon. This whole thing in there. And that R is supposed to be polar because it contains an electronegative element. You have different electronegativity in here, and that electronegative is connected to a hydrogen. We still have the backbone of the amino acid with the alpha carbon with the amino group to the left and the carboxylated group to the right for each one of those. And these are serine, threonine, tyrosine, tryptophan, cysteine, asparagine, and glutamine. Amino acids can also have a basic polar side chain or an acidic polar side chain. So for basic polar side chains, they should have a positive charge. And for acidic polar side chain, they have a negative charge. There are 3 amino acids that have a basic side chain at a neutral pH, and these are histidine, lysine, and arginine. Their side chains, they do contain a nitrogen. It's the one that I highlighted in there. And that nitrogen resembles ammonia, which is a base. Their pKa are high enough that they tend to bind with a hydrogen. So that was originally just a nitrogen in there. It binds with a hydrogen because it's very basic. It takes a hydrogen because we know that bases take hydrogens. So that one takes a hydrogen and becomes a positive charge. Same thing, this was an NH2. It takes a hydrogen and it becomes an NH3 positive charge. Same thing, this one was an NH. It takes a hydrogen and it becomes an NH2 positive charge. This is why amino acids that are that have a basic polar side chains, they should end with a positive charge. On the other hand, for the acidic polar side chain amino acids, they do carry a negative charge at a certain pH value. An example is aspartate or aspartic acid, or glutamate or glutamic acid. These 2 amino acids, they do have acidic side chains at a neutral pH. Their side chains, they do have the carboxylic group, as you can see over here. This is the carboxylic group, which we call it the carboxylate. Their pKa's are low enough to lose a proton. So that was originally an acid, a COOH, but it lost a proton because the pKa is so low. They lose a proton in there and they become negatively charged in the process. So all what you need to know is that amino acids that are acidic, they should have an R chain that ends with a negative charge, and amino acids that are basic, they should have an R chain connected to the alpha carbon that should have a positive charge. With respect to the amino acid stereochemistry, with a few exceptions, the amino acids that exist in the living system is always in the L form and not in the D form. The L form means that the amino group is located to the left. Okay, so it is located to the left in there, and not to the right. This is why we call this, it's the L amino acid isomer. Of course, it does have a mirror image, as you can see, because this carbon is a chiral center. So if I want to go ahead and draw the mirror image of the L, it's going to be the D. And the mirror image is going to be a CH and NH3 positive, and then the base of the acid, and then the R. and that is the D amino acid. Okay, it's always that the mirror image of the L is the D and the mirror image of the D is the L, but most amino acids, they exist in the L form instead of the D form. Now, almost all amino acids, they do have a chiral center. The only exception is the glycine. So this is why 19 out of 20 amino acids, they are chiral, meaning that they have a mirror image. They have a chiral center, they have a mirror image, and the chiral center, of course, is the alpha carbon. Okay, so that is going to be my alpha carbon in there. Glycine has no chiral center, so the alpha carbon in there is not a chiral center. And the reason why it is not a chiral center, because it is not connected to 4 different groups. So this means that this has no mirror image, because the mirror image is the same as the molecule itself. So this is why it has no mirror image.